So just to go over the goals and objectives of this presentation, first I'm going to talk a little bit about freshwater mussels and why they're important. I'm going to explain what environmental DNA is. And then I'll talk about my study specifically. So where I've been conducting this study, what my intentions are, and what I still plan to do in the future. But just a little bit about freshwater mussels. There were once around 300 species of them in North America, 2/3 of which have been extinct or they're in peril now. There are 67 known species in Pennsylvania alone. At least 10 of these are endangered. So there are a lot of species of mussels in North America specifically. They're a highly threatened group of organisms. And part of why they are vulnerable is because their life cycle also depends on a host fish species. So you can see on the left here is a life cycle of just a typical mussel. Fertilization occurs externally, and then inside the female mussels, the glochidia or larvae develop, and eventually that mussel, they have different techniques for basically injecting their larvae and having them attached to the gills of a fish. So they'll have specific hosts, and they'll attach their gills and stay on there for about 2 weeks until they fall off as juvenile mussels. So why are mussels important, and why are they threatened? So they provide several key ecosystem services. They're filter feeders, and so they also filter out a lot of harmful substances in the water, including pollutants and sediment. They create important habitat for fish and other macroinvertebrates, and also serve as food for some of those animals. And because they're highly vulnerable to pollution and water quality, they're good indicators of water quality. So they're threatened by various things, including invasive species, pollution, and habitat destruction. And so in these pictures, you can see 2 of the key invasive clam species that either compete with native freshwater mussels, or sometimes they'll even attach to their shells to make it impossible for them to feed. So here are just some pictures of freshwater mussels that I have found in the field. The 2 pictures of like the larger mussels are Lasmogona costata or fluted shell. All of these are found in Pine Creek. The smaller one is the lilliput, which is a little bit more rare. So now that you have an idea of what they look like, this is just generally what you might see if you were walking through a stream conducting a mussel survey. You just kind of see like this, sediment covered stream bed. If you haven't found it already, there's the mussel. You can see its opening right there. And so you can imagine that this might be hard to spot a lot of the time, especially if conditions are poor. So if there was just a recent rain event, it can wash a lot of silt into the water and make it even harder to see. And they're also just very good at disguising themselves as rocks. So yeah, it's hard to find them. In a study that was done by Cameo and Downing, they actually found statistically that in midsummer, around 18% of the mussels were completely buried, and in late fall, it increased to 60%. So whenever you're conducting a survey like this that relies on the site, there are a lot of mussels that you're not seeing at all. And so that's where eDNA can come in handy. So eDNA is DNA that's shed by aquatic organisms in many different forms, including tissues, feces, and gametes. And then, so the scientists can then collect water samples that contains this DNA and extract it for analysis. So this method has several advantages. It usually has equal or higher detection probability than morphological studies. It's less invasive because you do not have to actually pick out any of the mussels from their habitat. It's usually lower cost and sampling effort. You need a certain number of people to help with traditional surveys, and that's not true for eDNA. And it avoids some logistical constraints. So sometimes sites are just hard to access, or again, weather conditions might be bad. So it can help with those problems. So how do we analyze eDNA data once we have it? Or how do we prepare for an eDNA study? So usually the first step is to choose a highly conserved region of a gene to amplify. And a lot of times this is mitochondrial DNA because it's highly conserved between generations. So scientists have to design primers that then target a specific sequence in a gene that's common to a broad taxonomic group of organisms. So in this case, it would be mussels. And then once you have those primers and have sequences produced by them, you can compare the sequences you get with a reference sequence database to see what species are present in the water samples we're taking. So what are the sampling locations that I looked at in this study so far? So I've looked in Pine Creek, which is a tributary to the Allegheny River. You can see this is the Allegheny River down here. And then off of this there's a map of the whole watershed of Pine Creek. And then within there, this little red square is kind of containing the three main sites that I've looked at. So Pine Creek across from the Best Feed store, Pine Creek at the intersection with Montour Run, a tributary. And then I also searched in Willow Run, which is a small tributary of Pine Creek. So just a little bit about those traditional mussel surveys. So these were conducted as hand searches with 2 to 4 people, depending on how much effort was needed. And basically all this is walking upstream without disturbing sediment and collecting mussels as you see them. You span the width of the stream, and basically if you see a mussel, you just pick it out and measure its length and try to determine its sex if possible, and then return it back to the sediment and keep going. So at Best Feeds, this took about 2 person-hours. We sampled a length of approximately 55 meters. At Montour Run, we sampled between the intersection with Montour Run and then around the sewage treatment plant of Pine Creek. So that was about 235 meters, and we spent longer there. Interestingly, in fall 2020, a specimen of Toxoplasma parvus, or the lilliput, was found at this site. That's the only other kind of mussel that was found in Pine Creek besides Plasmodona costata. We did not find any lilliput in fall 2021. And then finally, Willow Run, we sampled about 71.5 meters of that tributary for about 2 person-hours. So just the basic results of these traditional surveys. So like I said, we found Plasmodona costata individuals at both Best Feeds and the mouth of Montour Run. We also collected some dead shells. That's what you see in blue here. And then at Willow Run, both years, in the fall of 2020 and 2021, we found no native freshwater mussels and very few corbicula, which is one of those invasive species I mentioned. Okay, so environmental DNA surveys, which is the kind of the second part of my project. So as I said before, you need to design a universal primer pair. And so the gene that we've chosen is WELT2, COX2, cytochrome oxidase 2, and cytochrome oxidase 1. So you can see here these arrows point to around the area where the forward primer is and the reverse primer. This yields a very large product. The primers have to be broad enough to detect many species of native mussels, but specific enough to omit non-target tags like those invasive species. And initially, we have to validate these large primers with tissue samples from mussel specimens we have. So we have 6 native mussels that we use their tissue DNA to kind of make sure that the primers are working. And so here are some preliminary results of that. These bands basically are showing the PCR products, the products of polymerase chain reactions, which is just making many copies of that segment of DNA we're targeting. And because they line up with this band here, this band is about 1,000 base pairs. It's because these are a little bit lower than that. They're around the size they should be. And there are no bands in the negative controls here. So that's also good, meaning we did not have contamination during the reactions. So the future directions for environmental DNA and the study in general is to take the products from those universal primers and use them to prepare something called a mock mixture. So those were, two universal primers that make a very large product. We also have 4 pairs of internal primers, and we will use that mock mixture to determine the most successful pair. And then we'll use that best primer pair to detect muscle DNA in water samples, so actual eDNA samples collected in Pine Creek and other places. And then in addition, we'll continue to conduct traditional muscle surveys in tandem with the eDNA so that we can compare the techniques. Compare eDNA results with traditional muscle surveys and see if eDNA is a viable tool to basically monitor for freshwater mussels. So just to reiterate why this is important and what we hope to accomplish, this fills a research gap in freshwater biology because still relatively few studies focus on freshwater mussels. It also contributes to a growing body of eDNA studies. There are a lot of studies of fish with eDNA, especially ones that compare electrofishing and eDNA. There are very few that do the same for freshwater mussel surveys. And finally, the results of this study will hopefully facilitate more efficient monitoring and therefore conservation efforts for mussels. So it's important to know where they are to prioritize places that need to be protected. And then finally, I just want to thank all the people that helped me with this research, my advisor, Dr. Brady Porter, for helping me, and then everyone who's helped me in the lab and with the field surveys. Yes, thank you. And I'll take any questions anyone has.
Hi, Meredith. It's Brooke. I was just curious why you chose the COX-2 and COX-1 gene.
Yeah, so the reason a lot of the eDNA studies focus on COX-1, specifically because it's a highly conserved mitochondrial gene, and also because within BLAST, which is how we compare the sequences that we get back from next generation sequencing to a reference database, a lot of the entries for that database are COX-1 and some for COX-2. So a lot of it is just by necessity that there aren't really available sequences for other genes yet, especially for mussels that haven't been studied as much. Does that help?
Nope, that's good, thanks.
Okay, if there's no other questions, we're going to move on to the next presenter then. Thank you so much, Meredith. So our next presenter is Rachel Partial, who's in Dr. Matthew Kostek's lab. And Rachel's presentation is Jo-1 protein induces autoimmune response and loss of force production, a myositis mouse model. Go ahead, Rachel.
Mary, if you could please enable host abilities or Victoria so I could screen share, that would be great. Thank you. Perfect. Can everybody see the poster?
Yeah, we can see.
Okay, perfect. Okay, so hello, everybody. I chose to do just keeping with the poster. I did not do a PowerPoint presentation. So what's going to happen is as I go through this, I will be zooming in and out of this poster as we go along. So let me zoom into the introduction here if I can. Okay. So hello, my name is Rachel Partial and I'm a physical therapy student here at Duquesne University. This research project focuses on creating a standard animal model for human myositis. We wanted this model to mimic the disease that is seen in human patients with myositis. There are currently no models of this disease, which hinders research. Myositis is a chronic autoimmune disorder which causes muscle inflammation and can be severely debilitating. There's no cure and the cause is unknown. Yet most patients develop autoantibodies against the Jo1 protein, also known as histidyl tRNA synthetase. Jo1 is one of the 8 tRNA synthetases linked to several anti-synthetase syndromes, which includes different degrees of myositis, inflammatory arthritis, interstitial lung diseases, and cardiovascular complications. The Jo1 protein triggers these immune responses, both innate and adaptive, which leads to cell-mediated damage to specific tissues, including the muscles and lungs. The inflammation at the level of the lungs occurs in the perivascular and peribronchial regions. In terms of muscle inflammation, it occurs in the perimysium, or the sheath that surrounds the muscle fibers. This specific pattern of muscle damage are commonly seen in people with myositis. The Myositis Disease Activity Assessment Tool, or the MDAAT, has also been used when comparing the associations between the presence of the antibody Jo1 antibody in blood samples and disease progressions for organ systems mentioned before, like the joints and even the lungs. Joint pain and muscle weakness is very common with this disease, which causes difficulty performing tasks of daily living. As a physical therapy student, I know that inflamed joints and muscles are difficult and painful to move. And as this disease process becomes worse, the less our patients will be able to do things in terms of hobbies, work, household tasks, like cooking, and even personal hygiene. So, like I said, the purpose of this study was to verify a myositis animal model using the Jo1 protein to mediate a pathologic immune response. So in humans, the muscle tissue shows no sign of recovery. And we also hypothesize that the intramuscular injection of the Jo1 protein in mice will mediate an autoimmune response similar to that seen in human myositis patients. Our experimental design compared saline, the control injections, to Jo1, the experimental group injections. All injections were delivered intramuscular to the gastrocnemius muscle. Muscle physiology testing and muscle tissue collection of the gastrocnemius occurred at 10 days, 21 days, and 42 days after injection. We wanted to determine when the muscle damage was going to occur after these injections, and if there was any muscle recovery after the longer time periods. Data from our preliminary trials did not show many signs of inflammation or muscle damage at the 10-day time point, so therefore we extended the time periods and added 2 additional time points. For our methods, we split the male and female mice into 2 groups. All mice were healthy prior to either the saline or the Jo1 injection, and their genetic backgrounds were not modified. As noted before, intramuscular injection occurred at day 0, and muscle physiology testing occurred at day 10, day 21, and day 42. Our muscle physiology testing was performed using the equipment shown in figure 5A. The mice were deeply anesthetized, and the sciatic nerve was exposed and stimulated at frequencies of 25 hertz, 50 hertz, 75 hertz, 100 hertz, and 125 hertz to develop a force frequency curve, measures of contraction velocity, and maximal force production. The foot was attached to a force plate right here, if you can see my cursor. I'm not sure if you can. And muscle force was generated by the nerve stimulation. Figure 5B shows the force production of graphs at 25 hertz of a mouse that received the Jo1 protein on top and the saline mouse on the bottom. As you can see by the staggered lines, a tetanic contraction was not reached, which is of course expected at this low frequency. Figure 5C shows the force production graph at 100 hertz of, again, a mouse that received the Jo1 protein on top and the saline mouse on the bottom. The smooth hump in the graph shows that a tetanic or maximal contraction was reached at this frequency. And I also noted in the poster that for the mice that received the Jo1 protein, they had a much lower force as compared to the wild type mice, 140 millinewtons versus 250 millinewtons. So then the muscles were collected, preserved for cutting, staining using H&E or hematoxylin and eosin, and F480 antibody stains to visualize and quantify macrophages. Microscopic images were then made to complete histological analysis. Okay. In the top middle section of this poster, you can see an example of an H&E stained muscle, human muscle tissue. Figure 3A shows normal healthy muscle. The nuclei are on the periphery of the cells and the fiber sizes are uniform. Figure 3B shows a perifascicular pattern of myonecrosis in someone with myositis. As you can see, there is a large presence of necrotic or damaged skeletal muscle that is being infiltrated by macrophages as an immune-mediated response. There are also centrally located nuclei, which is another sign of muscle pathology in these stains. For a person that has myositis, the inflammation usually occurs in these specific areas of the muscle as opposed to being more widespread, like would happen in response to acute injury or other muscle diseases. We also have H&E stain pictures from our experiment in the middle of the poster in figure 4. Figure 4A shows saline injected mouse tissue with nuclei on the periphery of the cells and uniform fiber sizes. Figure 4B, on the other hand, shows tissue of a mouse injected with this Jo-1 protein that is pathologic. The fibers have degenerated and there is a lot of inflammation present, primarily in the perifascicular region. In the top right section of the poster, figure 6, our data shows that the contraction velocity in milliseconds, which is one of our measures of muscle function. The female mice are represented in the orange colored bar graphs and the male mice are represented in the blue colored bar graphs. Our data showed that at successive time points, the muscle velocity decreased for the mice that received the Jo-1 protein. When comparing males to females, the female mice that received the Jo-1 protein had reduced velocity compared to the male mice. Underneath that graph, we have our second graph, figure 7, which shows muscle force production normalized to body weight. This is our primary outcome of muscle function. We found that the mice that received the Jo-1 protein had significantly lower muscle force production compared to the mice that received the saline. And this is statistically significant with a p-value of less than 0.05. We also noticed that peak muscle damage or loss of force production occurred at the 21-day time point. And this occurred for both male and female mice. Histological analysis is still ongoing, but the preliminary observations concur with the muscle physiology testing. So, in conclusion, injections of the Jo-1 protein triggers inflammatory processes and consequent muscle fiber damage that are similar to those seen in clinical myositis patients. Injection of the Jo-1 protein appears to be a good model of myositis that can be used to test multiple interventions. Our laboratory plans to replicate this experiment once more while increasing our sample size in order to verify the results. We are also planning therapeutic trials that would involve treatments including anti-inflammatory agents in order to help with the muscle damage caused by the Jo-1 protein. Now, I would like to take a moment to thank my incredible advisor, Dr. Matthew Kostek, and the intelligent and diligent women who work for the lab as well. It is an absolute honor to have had the experiences in the laboratory with you all this past year, and your efforts towards this project are greatly appreciated. We would also like to thank Dr. Dana Asherman for his assistance, advice, and production of the Jo-1 protein, which we used, and Dr. Paula Clemens for her assistance and advice on this project. Thank you so much for listening. Thank you to my friends and family for joining, and I will now turn it over to anyone who has any questions or comments.
Hi, Rachel, it's Brooke again. I have some questions. So it was a really, really nice talk. Is the Jo-1 protein naturally present in the body? And it's just like the immune response is like, you know, immune response gone bad and it's making a response to this protein? Or is it something that shows up when this disease occurs?
I believe it is something that shows up as the disease process occurs. I might not be 100% correct on that, but I'm almost certain that that's how that happens.
Okay. And then do you think that this difference where your females are seeing like a more extreme response than your males could be a difference in the immune response? Because females seem to have more autoimmune disorders. So maybe that could maybe be something like that.
that definitely could be a possibility, and that's a great thought on that. We also did different graphs comparing body size and muscle size. So we also might assume that the size of the mice might have something to do with that as well. But like you said, that also could be your first point. That could be very true.
Thanks.
Yeah, you're welcome.
I have a question. So you mentioned there were other proteins associated with these types of diseases, like in the same class as Joe 1. Have people done histology for like tissues with those proteins as well? And does that look similar to yours, or has that not been explored yet?
From the research that I have done, it does look very similar. And these up top, these human pictures were not from our lab, and they do look very similar to that. So the nice, normal, healthy muscle tissue and those beautiful little circle type shapes, and then just next to it, just a lot of the dots and purple and everything like that. And the perifascicular or peribronchial regions were kind of what they looked like as well.
I have a question. So after you finish this research, how do you think this information will impact physical therapy, or is that not something you'll find out until you do further research?
Great question. So I believe that once we get into these therapeutic trials where we have these nanoemulsions, or what they're called, with the anti-inflammatory agents, I think that will be a good marker to whether or not those work. But as we as physical therapy students know, I think it is important to try and do other things in these human patients as well, including, joint range of motion, keeping them active, avoiding contractures. So in terms of this research specifically, I am excited to be kind of dual, PT student and also being a part of this lab, because I get to see the clinical and also the real biological and medical, medicine specific therapeutic agents and stuff. So I think that in the time being, the things that we have coming up for the lab could help. But in the time being, keeping them as active as possible, keeping their joint range of motion and strength is super important as well.
And real quick, Rachel, I just wanted to ask what you felt the most challenging part of your research was.
Definitely. Hold on, give me a second. I'm choosing between two different
It's a very open question, yeah, but I'm just very interested in the whole process here.
Yeah, so as you can see down here, when we had the mice, the fully anesthetized mice set up in this situation, I think I would say the word, it was interesting, and probably my favorite part of being a part of this process as well was having that sciatic nerve stimulation. And we had it hooked up to the computer, and when we tested at all these different frequencies, making sure that Maddie, who was had the I don't know what it's called. It's the needle, basically, or metal pole that was lined up to the sciatic nerve, I think was a pretty difficult part of this experiment, keeping it on there while the force was going, and then making sure that we had all of the different hertz and contractions, making sure that we were doing each in successive order so that we didn't fatigue the mice. That was something really important as well. And if that did happen, obviously we took note of that. But I think in terms of setting it up, making sure the sciatic nerve was stimulated, that we had the metal right on the nerve, and making sure that was all set up, and making sure that the foot of the mouse on the force plate was set up very well, I think was probably one of the harder parts of this. But as we went on, we kind of got the hang of it.
Okay, thank you so much. Great work.
Yeah, thank you so much.
Want to see this?
Any other questions? Awesome. Thank you again, everybody.
Thank you, Rachel. So our next presenter is Brooke Deal. She's in biology here at Duquesne, and her faculty advisor is Dr. Pollock. And Brooke's project is sex differences in the efficacy of research by nanotherapeutic correlates to macrophage infiltration. So go ahead, Brooke.
There we go. So like Victoria said, I'm Brooke, and I'm from John Pollock's lab. And today I'm going to be talking to you about the differences in this nanotherapeutic relief correlating to macrophage infiltration. So I believe that everyone in this audience today has probably experienced some type of pain in their lifetime. Most of us are familiar with this acute nociceptive pain, which this man here doing a belly flop is about to experience. And this type of pain is temporary, and it serves the purpose of telling our body that something hurts and to not do it again, to prevent us from doing any type of damage to our tissues. And when you experience neuropathic pain, something like you stub your toe, it goes to your brain, and this happens over and over again. Sometimes sensitization of the nerves can occur where chronic pain happens. And chronic pain is pain that lasts, you experience every day or for almost every day for about 6 months. During a neuroinflammatory response, there's some crosstalk occurring between neuronal cells, glial cells, and immune cells. And a major focus on the immunological side has been on the macrophage. And this is because the macrophage produces these inflammatory cytokines, or PG2 cytokines, which sensitize neurons and causes neuroinflammatory pain. Working with Dr. Jay's lab here at Duquesne University, we have had the opportunity to use a novel celecoxib nanoemulsion to block the COX-2 enzyme from producing the inflammatory cytokine and thereby decreasing pain. And utilizing the macrophage's natural response is an actually really cool technique because the macrophages uptake or phagocytose the nanoemulsion in the peripheral blood. And then when they sense an inflammatory response, they go to the site of injury, delivering the drug there. So in this way, a macrophage acts as both a delivery system and the target. And my lab has done quite a bit of work in this. And in males, they have seen that a single injection has provided 5 to 6 days of sustained relief. But like many others in the field, until recently, females weren't assessed. And so, in fact, it wasn't until 1994 that the NIH started to require that females be used in clinical trials. And it wasn't until 2016 that they started to mandate the consideration of sex as a biological variable in preclinical research. And this means that a lot of our knowledge of basic science and preclinical research has been used, focused on male animals and cell lines. So there's a lot about the female inflammatory response that we don't know about. Current literature shows some key players in this immune response, in this neuroinflammation response. And like I said, the macrophage, but also the mast cell has been shown to be a main inducer in male inflammatory pain and also in female inflammatory pain. But the T cell is a new thing that recent scientists have seen to be a key player in inducing and maintaining chronic neuroinflammation in females. So keeping this in mind, to be able to study neuroinflammatory pain, we have to have a model. And our model in the Pollock lab is the chronic constriction injury. In this model, we tie 4 ligatures around the sciatic nerve to constrict the nerve and cause some inflammation. We can then probe the hind paw in order to test the sensitivity or the pain-like response. In this next photo here, you can see me probing the hind foot of a rat with what's called a von Frey filament. The rat's not bothered. His foot's flat. He's checking me out, but not too bothered by it. And these filaments come in different sizes. So a small filament like this, like a hair, or a big filament like this, like a piece of spaghetti. And the bigger the filament, the more grams of force that can be applied to the paw. Through this pattern of response and non-responses, we come up with a 50% withdrawal. So when the rat will pick up its foot 50% of the time. The data is then graphed on a graph like this with time along the bottom and grams of force along the side. And so the larger the grams of force, the less hypersensitivity or the least amount of pain the rat is in. And the smaller force that they're responding to, the more hypersensitive they are or the more pain they're experiencing. So I did these studies in male and female rats, and I performed my chronic constriction injury. And after the surgery, both males and females experienced hypersensitivity, dropping down here. And they both are pretty much equivalent to each other. So after the surgery, males and females are experiencing the same amount of pain. And it's not changed by giving them an empty drug-free nanomulsion. Once the drug-loaded nanomulsion is given to males, they experience this significant relief lasting about 5 days. And then once it is given to females, they too receive significant relief, but at an attenuated rate compared to the males. And in fact, there is a significant difference between the male and female relief. And because this drug is macrophage targeted, that was the first thing I wanted to investigate, was the level of macrophage infiltration at the sciatic nerve. So here you can see some green macrophages inside the sciatic nerve. And we have our controls, which is called a sham. And all we do is we open up and expose the sciatic nerve, clear it off, and close back up. And you can see that both males and females have little to no presence of macrophages in the sciatic nerve. And then after surgery, you can see that there's this infiltration of macrophages, and that it's not changed by the presence of drug-free nanomulsion. Once the drug is given, you can see this decrease in the number of macrophages present at the sciatic nerve. And visually here, you can kind of see that there are a greater relief in the males than in the females. So I quantitated this, and you can see here that there is a significant difference between the number of macrophages in males and females once they're given drug. So these samples were taken at the very peak of relief. And so this may explain the difference between the behaviors. One of the other things that I thought about was maybe there's a difference in the ability for female macrophages to uptake the drug compared to males. And so I looked at this because the nanoemulsion has this near infrared dye, and so we can visualize it like this. You can see it's purple, and females, it's about half the macrophages present at the site of injury that carry the nanoemulsion. So it doesn't appear to be that there's more or less being delivered to the males or females. Like I had mentioned earlier in my presentation, there's also another key player that the scientists in this field think might be involved, and that's the T cell. So I decided to look at T cells in the sciatic nerve, and you can see that in both males and females, and in all conditions, there's pretty much no change in the number of T cells in the sciatic nerve. And I found this interesting. And one of the things that I did discover while doing this was the localization of the T cells. So I evaluated them within the nerve, the fasciculated nerve, but the nerves are surrounded by a sheath called the epineurium. And as you can see here, these green T cells are sitting out there in the epineurium. And that's true for both helper T cells and cytotoxic T cells. So maybe the T cells aren't influencing this difference within the nerve itself, but maybe adjacently in the epineurium. So overall, I've been able to show you that male and female rats experience the equivalent mechanical hypersensitivity following chronic constriction injury, and that celecoxib nanomulsion provides a significant relief to both males and females, but they're not the same. That COX-2 inhibition does not provide equivalent relief to the females as they do to the males. And some possibilities behind this difference could be this difference when COX-2 is inhibited, that females have significantly more macrophages than males. It could be that these T cells are influencing the neurons adjacently rather than directly within the nerve. And one of the pieces of data that I haven't showed today is that overall, females seem to have a higher rate of immune cell infiltration than the males. So we're still investigating this. But I'd like to thank my advisor, Dr. John Pollock, my lab members, Katie, Bria, Alicia, and Charles, past lab members, my collaborators, especially Dr. J, who has supplied us nanomulsion, my committee members, and of course, my funding. So I will take any questions.
Hey, Brooke, this is Maddie.
Hey, Maddie.
Hey, really great presentation. That was really nice data. Thank you. I was just wondering, do you guys give the same amount of drug to both male and females?
So yeah, we give this like 300 microliters to both the males and females. So technically, if we're going off their size, like if we would have done like a concentration per, per kilogram or whatever, or per gram, the females are getting more because they're smaller. The females are about 50 grams smaller than the males at this age.
Okay, thank you.
Yep.
Brooke, I had a question. It's Dr. Kostick. First of all, thanks for excellent presentation. Really nice project as well. My question is about the COX-2 enzyme. Do we know if there are many differences between males and females, and whether the COX-2 might be more present in the macrophages versus the T cells, or just what the sex difference might be with COX-2? Do we know anything about that?
So there's a paper, it's published in 2006. It's Chillingworth, and I could probably email it to you afterwards because I know you deal a lot with this as well. But it shows that, at least what I got from the paper, was it looks like COX-1 and COX-2 seem to be additive in females, and COX-2 seems to be more of the controller in males. So maybe it's also that females have this, like something else going on in the background, that it's not all controlled by COX-2. But to answer your question, I'm not so sure if there's like a different amount within the macrophage in males and females or in T cells, but I do know that difference has been seen. They did like COX-1 and COX-2 knockout mice, and I can forward that your way if you'd like.
Wow, thanks a lot. Yeah, excellent. Excellent. That's really nice.
Okay, then if there are no other questions, we're going to move on to our last presenter, who is Claire Kreider. So Claire's a student in the Department of History here, and Claire's advisor is John Mitchum. And Claire's presentation is The Subsistence of the People, Climate-Induced Food Insecurity in Pre-Revolutionary France.
Hi, thanks for having me. Ooh. In 1788, Jacques Necker, finance minister for Louis XVI, wrote, "The subsistence of the people is the most essential object that must occupy the administration." This statement was founded on the uncontested policy convention held by the French government that social stability could only be maintained by guaranteeing a stable supply of grain and cereal. Incorporating archival research conducted at the British Library in London, England, meteorological observations, diplomatic dispatches, and grape harvest data, in my paper, I argue that in pre-revolutionary France, extreme weather episodes resulted in food insecurity and to increase political and economic instability in an already volatile and vulnerable state. These factors destabilized the country and aided the middle class in overthrowing the existing regime, thus commencing the French Revolution. On the eve of the French Revolution, the French economy was in a dire state. Years of war, overspending, ineffective taxation, and feudal privileges left the country in loads of debt, which placed most of the economic burden on France's poorest class, the sans-culottes. 95% of the sans-culottes' diets consisted of bread and cereals, and because such a large portion of their diets came from these grain products, they were extremely susceptible to the effects of weather, pests, and crop disease. To make matters worse, France witnessed a bizarre series of weather events in 1788 and 1789, probably owing to the result of a volcanic explosion in Iceland earlier in the decade, which spewed ash, dust, and sulfur into the atmosphere, significantly lowering the temperatures in France and the rest of Western Europe. There was a low yield famine following the extreme weather events in 1788 and 1789. When famine occurred, grain prices skyrocketed. After this inflation, the poor can now expect to spend at least 85% of their wages on bread alone. Bread riots began to break out in August of 1788. Although at the time, these riots were largely the result of famine and did not carry any anti-regime overtones. After the meeting of the Estates General met in May, however, calls for the end of the Ancien regime emerged in bread riots and public protests. France experienced high atmospheric surface pressure in the spring of 1788. The data suggests that France was under an anticyclonic circulation not conducive to precipitation formation. These conditions account for low rainfall and high temperatures in Paris and the surrounding regions. France experienced a hailstorm on July 13, 1788, that covered a large portion of central France and destroyed vineyards, wheat fields, livestock, and property with hailstones 16 inches in circumference. The western province of France also experienced a hailstorm in May and was in no position to recover from the later July storm. Additionally, Paris saw a rainfall deficit of 10% in 1788. Other nearby areas experienced deficits of 35%, and southern provinces experienced deficits of 85%. During the spring and summer of 1788, France experienced average temperatures that were roughly 20 degrees hotter than what they had been the previous years. The data suggests that low rainfall was aggravated by high temperatures and evapotranspiration, causing young crops to wither in the heat, resulting in crop failure. The winter of 1788 was particularly severe due to its duration. Paris saw 86 days where temperatures sank below freezing. In the spring, the thawing of rivers and streams caused waters to rise, flooding wheat fields and pastures and bursting through rudimentary retaining dikes. When crops fail or yields are lower than expected, less food reaches the market, and this increase in demand causes prices to rise. The data suggests that the wheat harvest of 1788 amounted to 7/12 the average. Additionally, the quantitative shortfall of the grain harvest itself was amplified by the low quality of the crops. It's commonly suggested that popular disturbances in France during the 18th century coincide with periods of high bread prices. The sources I've analyzed confirms that this is true. The price of wheat and rye at the beginning of 1787 was only slightly higher than the price of wheat and rye at the beginning of 1786. There's a notable rise in March of 1788 in anticipation of a poor harvest. And this rise grew steeper in June, and by January of 1789, prices of wheat were 50% higher than what they had been the previous year. The highest price occurred on July 14th, 1789, the day the Bastille was stormed. France periodically experienced bread riots throughout the 17th and 18th century. Owing to the dearth of grains, bread riots would usually break out in the late winter or early spring. However, the harvest failure of 1788 was so severe that the first bread riot broke out in August of 1788. It became obvious by March of 1789 that that year's harvest would be inadequate. Therefore, as the price of grain rose, so did the number and degree of violent riots. Accordingly, 300 violent riots broke out all over France during the months of April through July of 1789. Until the spring of 1789, the bread riots across France generally lacked any anti-regime overtones. Until the meeting of the Estates General, almost all riots were exclusively a consequence of the crop failure caused price increase of bread. The anti-regime overtones were further solidified on June 17, 1789, when the Third Estate reconditioned themselves as the National Assembly. After the meeting of the Estates General, these disturbances assumed a political message, and because the poor had no real political leadership, the bourgeoisie found an opportunity to organize the urban poor under a united call for affordable bread, the abolishment of feudal dues, and the easing of taxes. The bourgeoisie took advantage of the bread riots, which in most cases weren't relevant to their own political and economic aims. In late June of 1789, France submitted a request to Britain for 20,000 sacks of flour to be sent to Paris to alleviate hunger in France. Debates filled Parliament as British officials debated whether or not Britain should send the requested aid. Although many politicians were sympathetic to the revolutionary cause, others found it difficult to feel bad for France given the two countries' rocky relationship. Politicians ultimately believed that sending aid to France would threaten the well-being of Britain and its citizens, and decided not to send the requested aid, and placed an effective ban on any wheat exported out of the kingdom. This ban would remain in effect until the end of the Napoleonic Wars. Merchants caught smuggling grain to France would be fined, arrested, and in some cases, executed. The price of wheat in France momentarily went down thanks to wheat imports from the United States, but food insecurity would only worsen during the years following the original 1789 revolution. Britain at this time also experienced their own bouts of inclement weather and poor harvests, which saw rates of hunger and malnutrition grow amongst the poor. Parliamentarians and elected officials that voted not to send aid to France on the claims that they first needed to help their own citizens did not follow through on their promises when their own public cried out, leading to much hunger and suffering in the United Kingdom, which would only worsen during the 19th century. By examining British debates of whether or not the United Kingdom should send aid to France, we learn a lot about our own sentiments towards those experiencing food insecurity and other sufferings, and what we're willing to do to combat them. US lawmakers have often stated that we should help our own citizens before offering aid to immigrants, refugees, and countries in need. And more often than not, the US avoids the cries of its own citizens when they call out. If the United States, just as Britain in 1789, wants to make the claim that we should help our own first, then we should do just that. Even before COVID-19, reduced supply chains and chronic and acute global hunger were on the rise due to various factors such as civil conflict, socioeconomic conditions, and climate change. To make matters worse, the United Nations Food and Agricultural Organization estimates that the global population will surpass 9.1 billion people by 2050, at which point our current agricultural system will not be able to supply enough food to satiate the world. And as climate change continues to worsen, this timeline is only sped up even more. Today in the US and across the globe, food prices are more than what they were last month and the month before that. The IMF reports that food costs have increased 6.8% globally since December, which was 3 months ago. Food inflation isn't just occurring in a faraway country that in any other point in time we would have been able to dismiss and downplay. It's happening here, right now, in Pittsburgh. Food insecurity threatens the stability of populations everywhere. And to remedy food insecurity amongst vulnerable groups, just like the sans-culottes in pre-revolutionary France, we must first get to the root of the problem. So, in closing, I ask you, where do we start? Thank you. I'd especially like to thank my advisor and department chair, Dr. John Mitchum, my graduate advisor, Dr. Philip Stelzel, and the Barty family for sponsoring the Barty International Research Grant, which allowed me to study at the British Library this fall. I'd be happy to answer any questions at this time.
Hey, Claire, thanks for your presentation. Kind of where you left off there, like where do we go from here? I think that's a really compelling question. For you, as you've thought about that, or maybe what are some things that are kind of happening around the area that might be helping to answer that question that you've been aware of?
That's a great question. Thank you for asking. We have a lot of great local organizations that supply vulnerable populations with food. But I think the biggest thing we could do is transition to green renewable energy. Food insecurity is only going to worsen as climate change continues to worsen. I think people are really finally starting to see the increase in food going up. In the United States, I paid $10 for a carton of strawberries the other day. I think encouraging people to walk more, drive their car less, take public transportation, invest money into public transportation and renewable energy is a really good place to start.
Well said. Thank you so much.
Yeah, of course. Thank you.
Hi, I have a question as well. What areas of the United States are being most affected by this besides Pittsburgh, and related to that, what industries? You can also tell abroad as well, like internationally, but I was just curious if your research had extended to those areas as well.
I really haven't looked into across the United States, but I know just from past research and reading the news that it's globally. Populous areas such as New York, LA, Seattle are really getting hit hard, but rural communities are also being hit hard. Globally, the United Kingdom is going through really terrible food inflation right now, and other kinds of inflation. So I mean, it's globally, it's everywhere. It's not just Pittsburgh, it's the entire world.
Yeah, thank you. I figured that was going to be your answer, but yeah, thank you. I just wondered if any specific areas, but I guess we should all kind of keep our eyes open.
Yeah, thank you for asking.
Of course.
Okay, if we don't have any other questions, then we're going to wrap up. Thank you so much, Claire, and all of our other presenters. And thank you to everyone who had questions. And I hope you enjoy the rest of your day. Thank you.